Dr. Matt Kaeberlein is an internationally recognized expert on the biology of aging and has authored over 200 different scientific papers in the field. I met up with him at his office in Seattle, and we had a great talk about everything from how far science has come with longevity, what supplements are legit and which are just hype, the science of biological aging tests, the practical things he does for his health and longevity, and much more.
If you cured all forms of cancer today, typical 50-year-old woman gets 3 years of life expectancy. If you cure all forms of heart disease today, it's the same number. So you don't get big effects on lifespan by only fixing one disease. Nobody should make any change in their health or buy any product based on what Brian Johnson says. If you're metabolically healthy, I wouldn't take metformin for a potential longevity benefit. Metformin does not increase lifespan in mice. Again, that has been widely misinterpreted. Resveratrol is the most debunked anti-aging molecule in history.
Just one particular influencer who says that this is the first time in human history that death might be a maybe. What do you think about?
So here's what I would say.
Okay, so we're here with Dr. Matt Kaeberlein. He's a biologist who's well known for researching the aging process. And Matt, could you just introduce us very quickly in terms of what you're working on recently? Because there's so much to say about your
Yeah, I have, for better or worse, worn a lot of hats throughout my career and recently. So right now, my day job is as CEO of a healthcare technology company called OptiSpan. And I would say our goal at OptiSpan is really to create solutions that will allow as many people as possible to get as close as possible to their optimal health span trajectory, recognizing lots and lots of challenges associated with that, including the fact we don't really even know what optimal is. But having said that, I think most people, particularly in the United States, have some low-hanging fruit that they can pick to get themselves 10, 15 years of better health span. And so we want to really figure out how we can enable that. And so we have a technology component there, but also a clinical component where we're actually working with people to, from a medical perspective, to, in a proactive science-based way, help them get on a better health span trajectory. And one of the nice things is, I've before that was in, the research world as a professor at the University of Washington studying the biology of aging. And so I can take a lot of what I learned from that research and that experience and start to apply it in the context of the real world. I firmly believe, and I think the science backs this up, that the biology of aging is first of all, the greatest risk factor for nearly every major cause of death and disability in developed countries, and that there are real things we can do today to positively impact that biology to keep people healthier longer, and that is only going to get better as the research continues to develop, and we'll have more and more effective ways to target aging biology to improve health. So it's a super fun place for me to be. I've learned a lot, and I can still continue to apply, what I've done in the past. And then I also still have a role with the Dog Aging Project, which is I think is probably the largest open science study, or one of the largest open science studies in the world. And the goal of the Dog Aging Project is really to both understand aging in companion dogs or pet dogs living with their owners, and then to use that knowledge to increase health span and lifespan in dogs. And so, I'm a dog person. I've always had dogs. I consider, all of the dogs that I've had to be members of my family. And so I appreciate the impact that if we're successful at increasing lifespan and health span in dogs, the impact that will have on people's lives. And much of the biology of aging is shared between dogs and people. And so it will also tell us something about human aging.
Yeah, that's really interesting, because with dogs, I guess you'll be able to, not only is it very important for people, and then probably better for stress reduction, happiness, which is going to translate to greater longevity, but there's also a sense of, we can more or less observe intuitively a dog's lifespan, of course, because they just don't live as long as humans. So you kind of get more of a concrete feel of what's working.
And you can learn much more rapidly, right? I mean, I think that's the thing. If you think about, let's just say you've got an intervention, let's just say a supplement. You've got a supplement that you think is going to improve health and increase lifespan in people. We would love to test that, right? We would love to actually do a clinical trial and learn, is that hypothesis correct? But to do that in people is a 20-year plus experiment. With how rapidly dogs age, especially big dogs, you can do a randomized, double-blind, placebo-controlled clinical trial for lifespan in 3 years. If appropriately powered, you can do that. And so that's one of the things that's one of the parts of the dog aging project is clinical trial of a drug called rapamycin to answer the question, does rapamycin slow aging, increase lifespan, improve health span metrics in dogs? So, I mean, I think there are many reasons why dogs are an interesting animal to study aging. One of them being that they're rapidly aging. Another one being that they share our environment, which is something we can't study in laboratory animals. In fact, we try not to. We try to control the environment. And they're so important to millions of people, right? Our companion animals, that there's intrinsic value in being able to improve their health as well.
Have you noticed anything yet in terms of dogs living longer in a particular region compared to another region? Maybe, they're going through more or less similar lifestyle, but then just through the fact that they're in this high altitude region or low altitude region or something's different?
Yeah, good question. I don't believe that anything has been published from the Dog Aging Project on that question specifically. Certainly, so there are about 50,000 dogs in the observational part of the study right now. And they're from all around the United States. So at least within the United States, as those dogs continue to age and develop diseases, and some of them will die, there will be opportunities to ask those kinds of questions, and certainly many others around air quality, water quality, socioeconomic status of the owners. A little bit has been done there, but I think, the study really only started to enroll a large number of dogs back in 2019. Okay, so the first NIH grant was end of 2018, and then mid 2019 we started to enroll a lot of dogs. So that's 6 years, but we're just now starting to get to the point where a significant fraction of dogs in that observational study are developing diseases of aging and mortality. So I think we'll be able to answer those questions going forward. There have been a few things that are particularly interesting. One that got a lot of attention was around frequency of feeding. So we asked the question, dogs that were fed once a day compared to dogs that were fed more than once a day, is there any difference in their risk of having been diagnosed with about 10 different age-related diseases? I didn't think that was going to work, but it turns out that there was a huge signal where the dogs fed once a day were at lower risk for all 10. It was really striking, and I think 7 of them were statistically significant. So that clearly says there's a correlation between frequency of feeding and risk of having been diagnosed with age-related diseases. But it doesn't say there's causation. And as a scientist, I'm already my first response is, what's an alternative explanation? So one hypothesis would be time-restricted feeding in dogs slows aging, right? That's a possibility. An alternative hypothesis would be dogs fed once a day are less likely to be obese, and we know obesity accelerates a whole bunch of age-related diseases. It's also possible that the owners who feed once a day do something different, or they feed a different diet. So we have to start to now try to answer those questions. But it was a very strong signal and a very intriguing observation that then can help us generate hypotheses to go test in a more mechanistic way.
That's really interesting. I remember something in rats. Well, there was a fact about rats that if you do once a day feeding in rats, it's almost the equivalent of a human eating once every 3 days, something like that. Is there
sort of? Yeah. I mean, again, it's complicated. So in some ways that's true. If you look at the metabolic rate of a mouse or a rat, like an overnight fast in a mouse or a rat is roughly equivalent to a fast of 3 to 6 days in a person, right? So in that context, yes, but the circadian rhythm of a mouse or a rat is still a 24-hour circadian rhythm. So in that case, no. So it's not you can't make a perfect extrapolation like that.
Yeah, I see. Okay, interesting. So you had mentioned targeting the low-hanging fruit for longevity with OptiSpan. Is that so? mainly focus on things the average person can practically implement right away, or are you also getting into more niche things like some compound like calcium alpha ketoglutarate or things like that?
Right. So here's the way I would frame it. I mean, I think our focus first is always on the foundation, and largely that comes back to lifestyle. And I would also put sort of proactive preventative screening and biomarker optimization, right, in that bucket. But still, I think for almost everybody, the first lever you should try to pull is to get your lifestyle somewhere close to the optimal range. And again, optimal is a tough word because that's what we're striving for, but that scares a lot of people, right? Because they immediately think, I can't eat any of the foods I like. I have to exercise 8 hours a day. You know, we're not talking about that. But if you look at the diet and sedentary lifestyle and, you know, stress levels and low sleep quality and, you know, challenges around human relationships that a lot of Americans have, there are some low-hanging fruit there that anybody can implement to improve those things. So we think about 4 pillars of health: eat, move, sleep, connect. Nutrition, exercise, sleep quality. And then connect is both around, you know, your personal connection, sense of purpose, mindfulness, some people would say, and equally, if not more importantly, your human relationships, right? I think if you can get, you know, somewhere close to optimal in those 4, I really believe the average American gets 15 to 20 years of health span. Now, again, there are people who have sort of tried to dial those things in. I think everybody, including me, there are things I can still do to improve. But if you think about the average American, I really believe 15 to 20 years of health span most people can get, which is a big freaking deal. So that's where we start.
Also, because you're saying health span, you're not saying lifespan, right?
Yeah, I mean, lifespan is complicated. You can our healthcare system, which I refer to often as a reactive disease care system because we typically wait until people are sick. So our reactive disease care system is really good at keeping sick people alive. We've done a great job at that. But what we haven't done a great job at is keeping people healthy. So I think there are lots of people, and this, we can talk about the statistics here. They're kind of shocking. You will sometimes see, you know, references to how health span is declining in the United States, and they'll say the average American lives, you know, whatever it is, 10 years with poor health span. The number's way bigger than that. The CDC estimates that 60% of Americans have chronic disease. The median age in the United States is 38 point something, 38.6. So if 60% of people have a chronic disease and the median age is 38.6, the math is not too hard to figure most Americans are living 30 plus years with chronic disease. Now, do we want to say that health span is over when you have your first chronic disease? No, I don't think that's a good definition, but I think we can say sick span begins. And so let's talk about reducing sick span, pushing that chronic disease out as late as we possibly can. That's what I mean when I say most people should be able to get at least 15 to 20 years of improved health span or reduced sick span. And then you asked about alpha ketoglutarate. We could talk about specifically about specific supplements or medications. I will say, yes, we do what I would describe as, and I genuinely believe as a scientifically rigorous approach, use some supplements, some prescription medications. Certainly we do it in a reactive way. If somebody has diabetes, we're going to get them on a diabetes drug. If somebody needs to get their lipids down, we're going to use lipid lowering medications or supplements as well. Lifestyle is still always going to be part of that equation. But then in a preventative way, when there is reason to believe, and when our physicians in consultation with the clients believe that this is the best approach for that individual, we'll use things where maybe we don't have the double-blind placebo-controlled clinical trial proving this is a preventative strategy, but there's a lot of data out there, we believe, to support those sorts of preventative approaches.
I see. Would you say that the average person is, how should I phrase it? So I think that a lot of people would be really enticed by certain longevity practices if it was framed in the way of, if you do these particular things, you can still binge drink on the weekends, right? I think that's part of the motivation to want to look for special pills or supplements, because they feel like, oh, I can just keep doing what I normally do, but then I just add the pill and it's not as bad, and I don't have to change my lifestyle too much.
I think that's true for some people, for sure. There is nothing that I'm aware of where the evidence supports the idea that you can take a pill to make up for low quality lifestyle.
Okay.
And I would say, I think first of all, I think that mindset, if people have that, I think the goal should be to kind of reframe that mindset, right? Why do you want to go out and binge drink? Why do you want to, eat foods that are going to detrimentally affect your body? Yeah, I get it tastes good in the short term, but in the long run, you feel like crap. You look like crap. Sorry, it's true. And you're aging more rapidly, right? So I think it's partly, and the behavior modification piece is enormously difficult. And I'm being a little bit glib, because it is absolutely the case that there are addictive components or addiction-like components to many of these things, right? I think the idea that the high calorie, very tasty foods that have been engineered to induce a dopamine response have something like an addictive quality. We can debate, is it true addiction or not? It doesn't matter. It affects people like it's addiction. And that's hard to overcome, right? So I don't want to make it seem like it's easy, but I think kind of psychologically reframing away from, I want to do these things, to I want to be healthy and I want to feel good, and I want to be able to do the stuff that I like to do. And in order to do that, here are the steps that I can take, is a better psychological approach to getting there. And again, I think I've seen this over and over, and I've experienced this in my own life. And look, by no means am I perfect. Like I was a latecomer to really taking a strong interest in my health. But it's been my experience that when you actually start to implement what I would consider healthy approaches to the pillars, you actually come to want to do those things, right? Like I've gotten to the point now where I go out to eat 9 times out of 10, I actually would rather have a salad than a cheeseburger, right? So I think you can kind of retrain your brain to want to and prefer the healthier choices.
It's almost like retraining the brain to have a different aperture of focus of, okay, we're not going to focus just on the reward that comes in the next hour, but we're going to, we're also calculating how we're going to feel over the next day, the next week.
Yeah. And I also think there's a lot about what we believe, right? Again, if you believe that you are sort of destined to eat that, piece of cake or that ice cream at a birthday party, you're going to do that, right? And our brain is really good at sort of filtering what we perceive in the environment through that perceptive lens, and then that reinforces the behavior, right? So it's not, and maybe it's framing it as sort of long-term goals versus short-term goals, but I also just sort of feel like in many cases it's just about reframing the way you perceive even those short-term goals.
I see. Okay. And I just wanted to comment on what you said about the addictive nature of foods. I think that's definitely the case for sugar, or at least there's a habituation aspect, because I live in Japan and everything is just way less sweet.
Yeah.
And it's like everyone's palate is adapted to this lower level of sweetness.
Absolutely. I mean, again, I think the reason why I sort of said it the way I did was sometimes, the sort of more the more pedantic nature of especially people in the science world, they're going to argue about, is it really from a biochemical perspective addiction, right? And my point is, for this conversation, it doesn't really matter, right? It's a reinforced behavior, right? I agree completely. And I think what you're saying is that our palate, and that's just one thing that reinforces our behavior, will change over time. That's sort of, I was alluding to that by now my preference when I go out to eat, right? Absolutely. It's the same thing's true with salt. So, if you have for many, many years always put extra salt on your food, your palate is going to adapt to that, and you're going to want that. And you can retrain it the other direction. It takes a while, but you will absolutely retrain. Brussels sprouts, great example. I hated Brussels sprouts. I hated them. I don't know why. They're kind of bitter, right? Whatever. Now, when let's say I'm walking, through the buffet somewhere and they've got Brussels sprouts, I'm like, oh, Brussels sprouts. I love Brussels sprouts. So you can in fact retrain your palate to like certain things and to dislike certain things.
It's like the behavioral outcome is the same, whether or not it's technically driven by the dopamine, this or that.
Right. Exactly.
Yeah. Okay. And so there's a bunch of different things I want to touch on more, like sirtuins, rapamycin,
Sure.
and speed of aging clocks. But before we jump into any specifics, what could you tell us a little bit more about the Wormbot project? I thought that was really interesting and seemed really efficient and smart.
Yeah, happy to. So the Wormbot is basically a robotic technology coupled to AI that allows for what I would call massively high throughput longevity drug discovery. So the goal of building this technology was really to enable experimentation in the longevity space at scale. And what I mean by that specifically is the ability to measure lifespan predominantly, although we can measure some health span metrics, but predominantly lifespan across hundreds of thousands or millions of different interventions. And an intervention can be all sorts of stuff. It could be a genetic modification, it could be changing the temperature or the quality of the air. But for simplicity, let's just talk about drugs, because I think everybody understands the idea of why you might want to look for a drug that can increase lifespan. Maybe we're saying why lifespan. I think, again, we talked a lot about health span. Health span is really what we're most interested in, although living longer, as long as you're healthy, is a good thing as well. But from the perspective of targeting the biology of aging, the easiest way to know that you have slowed the aging process in a population of animals is if you can substantially increase average lifespan and maximum lifespan of the population. That's not true the other direction. There are lots of ways to shorten lifespan that have nothing to do with aging. But if you want to extend lifespan by a lot, you almost have to target the biology of aging. And that's because if you only fix one disease, you only get a tiny, tiny effect on lifespan at the population level. In fact, many people may not be aware of this. If you cured all forms of cancer today, typical 50-year-old woman gets 3 years of life expectancy. If you cure all forms of heart disease today, it's the same number. So you don't get big effects on lifespan by only fixing one disease. Why? Because all the other diseases of aging are still going up exponentially. So that's why we focus on lifespan.
Is it like you have one process that is happening that is increasing aging, and then you are getting all those risks at the same time, or do you have different types of processes that you have to target differentially to prevent cancer, heart disease, so on and so on?
Really good question, really complicated answer. I'll give you the simple version. It's both probably, but I think the important point is we know there is an underlying biology of aging that increases our risk for every age-related disease. And while that's complicated and involves a bunch of different processes. So people talk about the hallmarks of aging. There are 12 of them. There used to be 9. There'll be 15 in a few years. There's probably thousands of things that are happening in aging. Okay. And there's a network under that biology that we can modulate, and we can target a single gene and slow aging, slow the entire process. How do we know we're slowing the entire process? Well, we can robustly increase lifespan in laboratory animals up to, 65% is probably the top end from a single intervention, at least a non-genetic intervention in mice. That's a pretty big number. And when we do that, we seem to slow the onset and progression of every age-related disease and the functional declines across many tissues and organs and the molecular pathologies that go along with that. So it seems as though there's an underlying aging process where we can target one gene and have a big impact on that network. I think the differentiator is targeting a disease after it's happened doesn't hit that network. It only hits that one disease. That's why and again, I'm not at all trying to diminish the desire to cure people's diseases. Okay. My mom had colon cancer a few years ago. I absolutely wanted that colon cancer cured. That's really important at the individual level. But at the population level, it's minuscule. What we really want to do is target the biology of aging to keep people healthier longer and prevent all of those diseases at the same time.
I see. How, so two things. I wanted to ask you how close we are to making, say, a worm immortal. And then I know you had Sim Land on just yesterday.
Yeah. He was just here a couple of days ago. Yeah.
And he gave a presentation at Vitalist Bay, and he was talking about this Japanese man who, I think he's currently 116, and up to age 111, he didn't have basically any issues until he got a kidney disease. And then after that, he started getting all these other issues. So would that be, do you think what happened, what do you think is happening there? Do you think he has a particular gene that it was expressed in a way that, you know, you were talking about, you could target a particular gene and basically reduce or increase lifespan dramatically?
Right.
What do you think was going on with that particular person?
Right. So a couple things to say there. One, I am very skeptical that he had no problems to 110 or whatever it was. In fact, I am 100% sure that if you looked at his ability to do certain things, right? Walking speed, lifting weights, at 110, he was not as functional as he was at 40, right? So he still had declines. I think probably what Seam meant was he didn't have any overt diagnosis of chronic disease, which is still super impressive, right?
I might have misquoted. So.
No, I mean, I just think it's important for people to realize there is nobody who lives even into their 90s who is functionally the same as they were when they were 20, right? Okay. So having said that, when you look at centenarians, so those are people who live to be more than 100 as a whole, what you see is that they kind of fall into a couple of categories. There are people like that who seem to live very late in life in the absence of disease. So they have what we might think of as a more resilient phenotype. If they get hit by a stressor, they're more likely to bounce back. Those are also, in general, thought to be the people who can get away with poor lifestyle, right? And for whatever reason, the poor lifestyle doesn't cause them to have the same consequences as it would in the rest of us, you know, mere mortals, so to speak. The other class are people who just hang on forever with chronic disease. So it's not even one solution fits all in very long-lived people. But in those people who maintain health very late in life, they do seem to have a genetic makeup that protects them against the insults that would cause disease and kill the rest of us. The exact component of longevity in humans that is genetic is still a little bit up for debate. You'll hear numbers like 20% of human longevity is genetic and 80% is environmental. Those actually are probably wrong because they don't take into account the fact that in those populations who are now over 100 today, many of those people died early in life from what's called extrinsic mortality or things that have nothing to do with aging. So you take those people out of the equation and it seems to turn out that it's more like 40 to 50% of longevity is genetic. Still 40 to 50, 60 to 50% is environmental. So there's still a lot you can do, but genetics is probably important. And we know some of the genes that are probably involved, things like FOXO and things in the mTOR pathway. And what's interesting there is that those same genes affect aging in laboratory animals, right? Which suggests that the biology of aging, it's another piece of evidence that the biology of aging is probably pretty well conserved throughout evolution, at least among mammals, and probably all the way back even into simple invertebrates. These same genetic factors seem to influence aging across a very, very wide range of species. Now, you asked about immortality. So there's just a couple of things to say there. I mean, one is there's no evidence today that we understand aging biology anywhere nearly completely enough to accomplish even what I would call extreme life extension in humans. So I mentioned earlier, the best we've done in mice in the laboratory with a non-genetic intervention is about 65%. That's caloric restriction. And that was accomplished in the 1980s, early 1990s. We have not beaten that record in 40 years.
That was only caloric restriction? It wasn't combined with any...
No.
Wow.
No, it was 65% caloric restriction, which I don't know what that works out to in people, but it's probably like 700 calories a day. You know
what? That just seems like you would just kind of starve to death.
And right. And I want to be clear, I do not recommend caloric restriction in humans for a couple of reasons. Do I believe it probably slows the biology of aging in humans? Yes, I do. And in humans, it probably causes other negative consequences that will more than offset the benefit you might get from slower biological aging. So leaving out all the social and psychological stuff, which is real and big, we know that in people, maintenance of muscle mass, maintenance of bone density as we get older is really important. Mice in the laboratory do not die from fractures or frailty or sarcopenia. People do. And I think it's very, very unlikely that at that level of caloric restriction, people are going to be able to adequately maintain muscle mass and bone density. There's also all sorts of other problems around, temperature regulation, loss of libido, hormonal dysregulation so from my perspective, I don't think that's a great strategy, certainly not for a quality of life or health span.
Would you recommend any amount of caloric restriction, like 10% or 5%, something like that?
I mean, again, I think we don't know. I would say if you just look at the mortality data, so risk of death and also age-related diseases, being towards the lower end of the sort of normal weight range is probably about right in people. But that's a population sort of, it's kind of like using BMI. It works pretty well at a population level, but at the individual level, there's all sorts of outliers. And I will also say I'm nowhere near that. I'm towards the higher end of the normal weight range. So
I see. I had seen a study or two there's at least one in primates suggesting that you don't get the benefits from caloric restriction unless the control group is eating excessively or eating unhealthfully.
Yeah, so that's a little bit of a myth. So again, the primate studies are complicated. There's really only been 2. Those were both in rhesus macaques. And neither of them, I mean, this isn't really a criticism of the way the studies were designed or the people involved, but neither of them were very rigorously designed just simply because of the nature of how hard it is to get access to rhesus macaques and do those studies in a controlled way. So, and they got sort of differing results. So the study at Wisconsin showed an increase in lifespan and reduced age-related diseases sort of across the board. Those mice were more, they had higher body weights and were fed a higher calorie diet. The other study didn't see an increase in lifespan, and the effects on health span metrics were less pronounced. That was at the NIA. Those monkeys were leaner, and that's where this idea you were referring to comes from. Yeah, if you're fat, you get a benefit from caloric restriction. But that study also had a lot of problems. The monkeys were all sorts of different ages when they started. And so if you start caloric restriction later in life, it can actually cause detrimental effects. So I would be cautious about that interpretation. If we go back to the mouse studies where these were done really well, and that study that I alluded to, it's from Rick Weindrich and Roy Walford. I can give you the reference. You can put it in the show notes if you want. They did a gradation of restriction all the way from what's called ad libitum, so the mice can eat as much as they want, to like 10%, 20%, 30%, all the way to 65% restriction. And in most of those studies that Rick Weindrich was involved in, they actually used the 10% restriction group as the control. So they didn't use the fully fed group. And what you see is a graded response where you continue to get larger and larger lifespan extension all the way up to the greatest degree of restriction they did was 65%. One of the interesting things, most people, the scientists in the field who are paying attention know this, but outside of that, most people don't know this. None of the studies being done today are anywhere near that magnitude of effect. And even people who are doing caloric restriction typically will do 20 to 30% restriction. The reason for that is that it's very difficult to get the animal approval committees to allow you to restrict 50, 60%. So even though that's where you get the biggest effect on lifespan, we actually can't study that today because of the regulatory environment.
Oh, I guess just the ethical hurdles alone would suggest that doing 60% calorie restriction probably would not be worth it in terms of you're not living a life you want to live.
Here's what I would say about that. I would say the goal of research in mice is, sure, it's partly to understand whether or not a particular drug or a particular intervention might also work in people, but it's also about understanding how is that intervention improving lifespan and increasing health span from a mechanistic perspective. Because the better we understand those mechanisms, the better our ability to go in and develop more targeted interventions that may have fewer side effects. Again, caloric restriction at that level, because of the side effects, is really, I think, a non-starter in humans, at least at the population level. But if we could understand, 65% caloric restriction is increasing lifespan because it inhibits mTOR and activates AMP kinase and does 4 or 5 other things. It actually probably does 10,000 other things, but there might be 4 or 5 that are really important for the longevity benefits. Then maybe we can go just do those 4 or 5 things without all the other stuff that caloric restriction is doing to get the effects. This is getting back to what you asked about before. Could we someday get to a magic pill where you wouldn't have to calorically restrict, but you could still get all the benefits? I think that's a heavy lift, but I think we can get to interventions where you can get some of the benefits.
Okay. How are you feeling about, is there a pretty clear signal in terms of mTOR is that kind of the key thing going on here? That suppression of mTOR is getting the most bang for your buck in terms of mechanisms worth exploring?
Yeah. So that's a good, interesting question. So one thing I would say about mTOR is part of that network that I talked about that sort of underneath the hallmarks of aging, this network of proteins and metabolites and interactions. And the way I think about mTOR is it's a node in that network that it turns out it's very amenable to tweaking the network in a way that affects lifespan and health span. Meaning we have a drug where we can sort of titrate how much mTOR is turned on or turned off. The drug is rapamycin. And when we do that, what we see are pretty big effects on lifespan and health span metrics. Is it like the master regulator of aging? I don't think so. I wouldn't frame it that way. I would just say it's a very useful node in that network. mTOR is a nutrient sensor primarily, although it senses other things in the environment. So when food levels are high, mTOR activity goes up. When food levels are low, mTOR activity goes down. Rapamycin is a drug that turns down mTOR, so it's kind of like tricking the cell into thinking there isn't much food around. So one obvious hypothesis is that the reason caloric restriction is increasing lifespan is because it's turning down mTOR. I think that's certainly part of the story. Caloric restriction does 10,000 other things too. So there's other stuff happening, both good and bad. With rapamycin, the biggest effect on lifespan that has been achieved in mice is about 25 to 30% lifespan extension. Caloric restriction, 65%. So we have not been able to get the full benefit of caloric restriction at any dose of rapamycin yet. So I would say it's part of the story. But it's not as effective as caloric restriction.
Have there been any studies that do say, if you have a mouse that is doing 30% caloric restriction plus rapamycin, then it equals, have they got?
Yeah, I think there's only been one study that looked at that, and it was really sort of inconclusive. And those studies are also hard to interpret from a mechanistic perspective. So this might get in the weeds a little bit, so you can cut it out if it does. Sure. But it's a concept that again, even people in the field who should understand this don't, right? So let's just say you've got caloric restriction and rapamycin. Okay, 2 different ways that we can affect longevity. So, and let's pretend we don't know anything about mTOR. You just got these 2 things that can affect longevity. You put them together and you see that when you put them together, you get twice the effect increase in lifespan that you get from either one alone. Okay, so there's one interpretation is those things are acting through different mechanisms. That's why you get this additive effect. There are 2 different mechanisms that increase or that affect aging. You've taken care of both of them, you get an additive effect. So that from a genetic perspective, we would say they're acting in different pathways, right? But the alternative is the dose that you've used of caloric restriction or rapamycin was suboptimal. They're both suboptimal doses. They're actually acting through the same mechanism, but you're using suboptimal doses of both. When you put them together, you've gotten closer to the optimal dose, and that's why you get an additive effect. So the only way to be able to interpret that experiment is if you have optimized both of them, and then you combine them, which we could do, except you can't do 65% caloric restriction anymore. So you can't actually do the experiment that you're suggesting in the way that might allow it to be interpreted. So that's a long-winded way of saying we don't know. And I don't think we ever will know exactly the outcome of that experiment.
Is it Because I asked you about immortality.
All right.
And is it kind of like you have to have this balance between anabolism and catabolism, and at some point, if you do too much, how do I put it, mTOR suppression, then the thing just can't live properly.
Yeah. So I don't think that has anything to do with immortality, but that is true. So again, I think the thing to appreciate, everything has a dose response, right? Oxygen, water, right? You can kill yourself with either of those things if you really wanted to. Caloric restriction, great example. 65% caloric restriction in mice is maybe the optimum. You go to 100% caloric restriction, that mouse is dead, right? So everything has a dose response. Rapamycin has a dose response. mTOR has a dose response. If you turn growth signals down too low, you can't survive. If you turn growth signals up too much, you can't survive. You get cancer. You'll get hyperplasia or hypertrophy, right? You're going to die. So it is a balance, but where the balance is optimal for longevity, and even more in the cases of like anabolism and catabolism, you really need cycles of growth and recovery. We really don't know. And it's going to be different in different tissues. It's going to be different in different people. It's going to be different in you when you're 20 compared to when you're 60, or if you're living in Japan versus the United States, the environment, all those things are going to influence sort of what is the optimal cycling of these growth and no growth signals. So I think kind of the best we can do right now is to say, this is sort of a global, crude, but I think accurate statement. In most developed nations, most people are overfed, or they get too many nutrients, and they're too sedentary. And so that combined leads to a situation where growth promoting pathways are sort of chronically elevated over what's optimal for health. Interestingly, and this is again where there's a lot of confusion, people sometimes try to use this argument about, well, we should just go back to, the way our ancestors lived 10,000 years ago. Because if it was good enough 10,000 years ago, that's going to be optimal for health. I think the thing people miss there is that, so that the idea is humans have evolved to live in a certain environment, and that's going to be optimal for longevity, right? Because we evolved for that environment, right? The problem is evolution doesn't optimize for longevity. Evolution optimizes for reproduction, period, full stop, or survival of your offspring, right? So in fact, evolution, the optimal thing from an evolutionary perspective is usually suboptimal for longevity. There's this trade-off. If you're optimizing for reproduction, you get actually less longevity. So that idea doesn't work either. And there's really no merit to the idea that we should try to go back and live like our ancestors did 10,000 years ago. It's going to be somewhere in the middle. There's a lot of stuff in our environment today that's killing us, for sure. There's a lot of stuff in our environment that's keeping us alive longer and helping us. So what we really need to do is figure out, how do we get more of the stuff that's helping us and less of the stuff that's killing us?
So basically, the achieving a lifespan that is like 130, 140, 150 is a very unnatural thing. So you have to kind of go out, even if the argument is that's so natural, it must be good for you. Yeah. It's like, well, what's natural only gets you so far.
That's right. So, right. So, we as human beings have not evolved to live to 100 years old, right? And one way to think about this is, let's go back 10,000 years ago. Most people didn't make it into their 40s, right? Most human beings didn't live into their 40s. A few people, 5,000 years ago at least, you can find records of people who made it in their 90s or 100s. So maximum lifespan potential hasn't changed in humans, probably, but average lifespan has changed a lot. And so most people didn't make it past 30. All of the selective pressure from evolution was acting to cause people to have children before they died, right? And to optimize that, the reproductive potential. Once you get past, 25, 30, the selection for you to be alive another year goes down dramatically every year. We have not evolved to stay alive. So most people believe that aging, what we know of as aging today, is really the absence of natural selection. We have not evolved to prevent aging. It could happen, but we haven't evolved that because we didn't need to. because we were done from an evolutionary perspective. As soon as we had kids and they made it to the point where they could survive on their own, you're superfluous, right? You don't matter. so that's kind of, I think, the most likely explanation. There might have been a little bit of value to keeping people around, particularly women around, to help raise grandchildren, but that value is tiny, tiny, tiny from an evolutionary perspective compared to just getting to the point where you have kids.
Right. Don't women typically live longer on average than men?
They do, yeah. They live longer than men on average, and that's seen, it's not only in humans. And we don't really know the mechanisms. And there is a growing, I think, I don't know if it's quite to consensus yet, but opinion in the field that women live longer, but probably not healthier than men. In other words, those extra years are not extra years of health span in general. It's really complicated though by menopause in particular, and with the Women's Health Initiative sort of debacle, I would say, in my opinion, that caused many women not to use hormone replacement therapy for many years. We know that has a negative impact on at least many health span metrics. So I think the question would be, if hormone replacement therapy could be done optimally for most women, would a lot of that apparent discrepancy in health span go away? We don't know the answer to that. My guess is yes.
Interesting. There's this one particular influencer who says that this is the first time in human history that death might be a maybe. What do you think about?
So here's what I would say. Again, I mean, there's nothing about the biology that says it's impossible, says that immortality is impossible just from random chance, but not aging. There's nothing that says that we can't dramatically delay or potentially even stop the biological aging process. So it is theoretically possible, just like traveling to another galaxy is theoretically possible. There is nothing, absolutely nothing at all in the literature or on the horizon that suggests that we are close to accomplishing that. And again, I think one piece of evidence, and these people who, want to be futurists, great. Futurists are great. It's a great gig if you can get it, because you can make predictions, and if they don't come true, you're dead. So it doesn't, you don't really get proven wrong. Okay. But they never answer this data, right? The data are that the best we've done is 65% increased lifespan in any animal, and that was done 40 years ago. And every experiment being done today is less than that. If we were really getting closer and closer and closer to longevity escape velocity, or whatever you want to call it, shouldn't we be getting better and better and better at increasing lifespan? That's not happening. And if anything, it's stagnated. So I'm actually quite bullish on the idea that, and I want to be clear, we've learned a lot about the biology of aging. We just haven't learned how to get past that, what seems to be a barrier. So I'm pretty bullish about the idea that we can take discoveries in the field of geroscience and apply them in people to get maybe most people out past 100 years, right? I think that's pretty realistic. But I have seen nothing yet that makes me think we're going to get most people out past 140 years, let alone live, 300 years, 500 years, 10,000 years. So, it's, I put those kinds of claims in the realm of religion, right? It requires faith. If you want to be in a religion, feel free. You can believe that. If you want to be a scientist, then you've got to deal in data.
I see. The funny thing is that particular person is also starting a religion, so it kind of makes sense.
Right. And I mean, again, look, I like optimism. Honestly, I'm perfectly fine with that being the goal, right? If the goal is to dramatically increase human lifespan, as long as we do that and also increase health span, great. That's a good goal. So let's start actually taking some steps in the science to get us there. That we started, earlier talking about the worm bot. That was actually my whole rationale for wanting to develop that technology. This recognition that the field has stagnated in our ability to increase lifespan. That's what led me to create that technology, because I really believe the reason why we've stagnated is because everybody's focused on this very narrow set of hallmarks of aging. Nobody's doing true discovery science to find other to figure out what we don't know, right? And so I thought one approach to doing that would be to take a step back. Instead of assuming that mitochondrial dysfunction or epigenetic dysregulation or telomere shortening was the thing we should target, let's take a step back and let the biology tell us what's important. Let's just look at everything we can possibly look at, test every molecule we can think of to test. And again, obviously, we can't test everything. That's too big of a number, but we can get up into the millions. Like, that's doable. Let's test millions of things, find the things that work the best, and then we can go back and figure out how they're working. And that's great because you win twice. You've got something that works way better than anything you know about, and you've got a new lead on understanding the biology of aging. So that's one of the ways I think we get to better than 65% effect on lifespan. There are other strategies people are taking, like looking in these interesting animals that seem to have delayed or even what some people call negligible senescence, right? So they don't show the exponential increase in mortality that most animals do.
Like a Greenland shark or something like that?
Yeah. So there are a few of these things. I mean, the hydra is one, people point, the immortal hydra. So naked mole rats are super interesting. So they're very genetically related closely to other rodents, rats and mice. Rats and mice in the laboratory will live about 3 years. Naked mole rats will live 30 plus years on average. The interesting thing there, and again, I would say there's not 100% consensus on this, but it's at least been proposed that naked mole rats have a more or less flat mortality as a function of age, meaning in people, our risk of dying, especially once we get above 40, goes up exponentially. And then it might plateau a little bit out at very late ages, but it essentially goes up exponentially. The naked mole rat, it doesn't seem to do that. So there's something different about their biology of aging that if we could get human mortality to flatten at a low rate, not a high rate, that would be great. Then people potentially would live much longer. So if we can understand those mechanisms, there's value there. So that's like, a different type of approach. Instead of using defined laboratory animals, yeast, worm, mice, flies, trying to study other animals that are sort of exceptional agers and understand their aging. So people call that comparative biology of aging.
Are there any interesting mechanisms at play with the naked mole rat? Like, do they have some kind of amazing antioxidant defense system?
Yeah. So, I mean, a lot of people have studied this. Shelley Buffenstein was one of the first people to really lead efforts here. Vera Gorbanova at Rochester is another. A few mechanisms have been proposed. I think the answer is we still don't know. So one thing we do know is naked mole rats are very protected against cancer. So they've got pretty advanced cancer surveillance mechanisms. Now, as we also discussed, that's not going to get you from 3 years to 30 years, right? You wouldn't expect only fixing cancer is going to do that. So there's got to be other things that are going on there as well. And like I said, a few things have been proposed, nothing that I'm convinced is particularly important. So I won't get into those mechanisms because I don't really believe that they're the key.
Speaking of like tracking aging and tracking any progress we're making towards the slowing down of aging, what do you think about these methylation tests, these speed of aging tests?
Yeah. So maybe first just kind of definitionally describe what we're talking about, right? So DNA methylation is one category of molecular feature that sort of go under this bucket of epigenetics. So people may hear these referred to as epigenetic aging clocks as well. That's just one type of epigenetic change. And epigenetic just means on top of the genome. So our genome is our DNA, AGCT, right? And then you can have these modifications on top of the DNA that influence which genes get turned up or turned down. And methylation is one type of modification. So there are hundreds of thousands, maybe millions of these places in our genome that can be methylated. And what people have observed is a couple of things. One is these methylation changes influence how cells function. So when we're born, we have what are called pluripotent cells. They can turn into any cell type in our body. A fibroblast for our skin, a neuron for our brain, a cardiomyocyte for our heart, all the different cell types. So they're pluripotent. It's epigenetic changes that influence that progression and tell that cell to become whatever cell type it's destined to be. So these things called Yamanaka factors, Shinya Yamanaka, who won a Nobel Prize, discovered them, are epigenetic modifiers that cause that to happen. And if you put those Yamanaka factors into a fibroblast or a neuron or a cardiomyocyte, you can reprogram them back to a pluripotent state. And when you do that, that seems to reverse at least a lot of the damage that those cells have accumulated with aging. So that suggests that epigenetic changes might be important for aging, and epigenetic dysregulation is one of the 12 hallmarks of aging. It also turns out if you take an old mouse and you do what's called partial reprogramming, you can have a little bit of an effect on aging. So you can partially reverse some functional declines, and so far, the best people have been able to do is about a 20% effect on lifespan, so not quite as good as rapamycin, but the potential is there that you might be able to do better than rapamycin someday. So that also supports the idea that epigenetic changes are important for aging. So other people, starting with Steve Horvath, looked at these methylation patterns. Again, remember, hundreds of thousands of these things in your genome, and they asked a mathematically pretty easy question, which is if we restrict ourselves to a few dozen, maybe 100 of these, which 100 are most strongly correlated with, and they started with chronological age, so how many birthdays you've had. So they took a bunch of people and they asked across all of these epigenetic or methylation marks in the genome, which 100 are most correlated with the age of the person, the way they change. And they found them. And those were the first generation methylation clocks. So you can take those same 100 marks in you, in me, in a random person off the street, and based on which marks are methylated, which positions are methylated, predict within a few years that person's chronological age. Very cool. Not particularly useful. Most people know their age, but still very cool. But then they had this, they made this observation that when you do that, there are a subset of people that are way off, either way above or way below their predicted chronological age. And they hypothesized maybe those people are aging biologically faster or slower. So they went back and created these second generation clocks, which are a different 100. It's not 100. I'm just saying 100. But it's close to 100, a different 100 specific positions in the genome that are methylated, and asked which are most associated with risk of dying in 3 years or 5 years or 10 years. And they were able to do this because there are these what are called longitudinal studies where blood was collected from lots of people 20 years ago, 10 years ago. Some of those people have died. And so we can use those biobank samples and identify the 100 best methylation positions that are correlated with risk of dying or risk of mortality. That's the methylation clocks that are talked about as predicting biological age. The idea being that higher risk of dying in 3 years it means you have a higher biological age now. I think the important thing to appreciate is risk of dying is really important. That's a good thing to know. It would be nice if you knew what to do about it, but still it's good to know. But there's an important thing for people to appreciate. That's not biological aging. None of these biological aging clocks are actually measuring biological aging. They are all measuring something that is correlated with biological age. Risk of death, risk of developing cancer, whatever the health metric is. So and it's a correlation to that thing. It's the 100 methylation marks that are correlated to your risk of dying in 3 years or 5 years. So all of these things are really a correlation to biological age. So it's just important for people to appreciate. None of them are measuring biological age.
So to put that in context, let's take the example of the man who's 116 and he was like disease free, or he didn't have a diagnosis until age 111. Yeah. So would that be kind of the picture of the person who has a very low biological age? Like his risk of death is low because he doesn't have a diagnosis, but he's still experiencing, a decline in his walking rate, reduction in muscle mass, something like that.
You would expect. Yeah. Okay. I mean, again, part of the challenge with biological age is it's a really good concept that we can't measure, right? And I don't even know that there's a consensus definition on what we mean. So yes, conceptually, people who are exceptionally healthy for their chronological age have a younger biological age, and vice versa. People who are exceptionally unhealthy for their chronological age, for how many birthdays they've had, are biologically older. But that's not perfect, because again, health is not 100% equivalent to biological age. There are lots of ways you could leave this room right now and 10 minutes from now be less healthy than you are right now without accelerating your biological age, right? So it's an overlapping sort of set of Venn diagrams, maybe is a way to think about it. But yeah, that's the idea that certain individuals are aging biologically faster than the average population, and over many years, their biological age is going to be different, higher in that case than their chronological age. And these tools can potentially measure that. But I like to say, again, it's a correlation to a correlation. I think these clocks are pretty good at estimating health, estimating risk of dying in the research setting. They're really powerful tools in the research setting. I think where they have the most utility potentially is if we can get to the point where we can say, and the reason why they're correlated with that is because these specific genes are being turned up or turned down due to the change in methylation. So that gets us to a mechanistic connection. So far, we haven't been able to do that. I think the other thing that's really important for people to appreciate, though, is, is these tests have been rushed to market. So there's been this rush to start selling these things to people direct to consumer. There's no regulation, no validation. None of these tests have been approved by any regulatory body or any independent body. And there's no transparency on the accuracy or precision of these tests that the companies are selling. So people have started to do their own experiments. I did an experiment. I tested 4 different companies, did duplicates of each test. I was 53.75 years old on the day I did that. It was the same biological samples, same day, sent them back. The 8 tests ranged from low 40s to mid 60s, right? I was 53.75 years old chronologically. If I average all of them, it was pretty close to 53.75 years old, but biologically it's nonsense. It's noise, right? I'm either 43 or I'm 65 biologically, depending on which test I want to pick. You know, if I was an influencer who's trying to sell you how great my protocol is working, I'd pick the 43-year-old test, right? And I'd say, I set the world record for, reducing my biological age by a decade in 3 months, right? But the problem is, when you actually look at all of the data, these direct-to-consumer tests, I think, are largely noise. I don't think they're actionable, but I also don't think they're accurate.
Okay, so if there's that much variability, then let's say someone has the means to run a test over and over again, and they're doing a competition to see who has the slowest speed of aging based on these tests, then that person could just kind of keep running the test until they got a lower score and then post that.
That's called cherry picking your data. Yeah.
Okay.
And it happens. I mean, we know that happens, right? Again, this is why I think if you're really a scientist, you do the test multiple times and you show all of the data together. So you get a feel for what's the variance, right? Statistically, what's the variance for the same sample on the same day? That's called technical replicates. And then you can do biological replicates where you would do it from different samples on different days, right? But yeah, I think you're right. That's exactly what has been happening. And again, I think this is the difference between an influencer on the internet and a scientist, right? In the scientific world, if you do an experiment and you get a set of data and you leave some of the data out because you're trying to convince the reviewers of a certain, model, that's called fabrication of data. That's the worst thing you can do as a scientist, right? That would not be accepted. And when people do that, they get in trouble in science. Unfortunately, on the internet, that stuff is just rampant.
Yeah. Okay. So more specifically, Brian Johnson said that he got a speed of aging test, a result of 0.48. I don't know how much you put into these Dunedin pace of aging scores, but what do you think about that? Is that particularly impressive or?
Right. So again, here's what I would say. So the Dunedin pace of aging comes from a really nice study looking at a different set of methylation marks. So first, maybe let's talk about what that's actually measuring, right? So the idea is you've got these what are called longitudinal studies where they've measured the same people over many years or decades. And those people had changes in health over that time. And we can use a variety of different measures to estimate how fast people's health is declining as they're getting older. And in that study, they found a different set of methylation marks. So it's a different pattern that was strongly correlated with how fast people's health changed as they were getting older. That's what the Dunedin pace algorithm is using. So again, in that study, in that research setting, in that population, those particular methylation marks were pretty well correlated with change in health over time. Is that actually measuring rate of biological aging? No, it's a correlation to rate of biological aging. It's a change in health over time. So it's a correlation to a correlation, but it's pretty cool and potentially useful. Why is it useful? Well, it's really useful if you can show across many different populations that it works the same. And if you can show that interventions that we are pretty sure slow aging, like going from a crappy diet to a high quality diet or going from sedentary to active, that we can show those things reduce the pace of aging, it starts to be convincing that this might be useful in people to predict whether or not rapamycin slows your rate of aging. You could start taking rapamycin and see if your Dunedin pace of aging score is lower. That would be super powerful. And that's a fantastic goal to have. One of the reasons why it's a fantastic goal is right now, it's really hard to do clinical trials to test a drug to see if it's affecting aging. If we had a test we actually believed was really good at telling you your rate of aging, that would help for those clinical trials. We don't have that. Dunedin pace is not that, at least as it's being deployed right now. But it's still, I think, an advance in the research world, and it will continue to get better. Again, the problem is this, when it's brought to the consumer realm, even with all the limitations that we've got in the research world, as soon as it gets brought to the consumer realm, we have no data on precision, accuracy, quality control. So again, I can tell you my data. So I did this. I did longitudinally for 2 years. There were probably 6 or 8. It wasn't, 50 tests, but that's a pretty good density of tests over a 24-month period. So my biological age bounced, 20 years up and down over that period, right? It's kind of funny. I actually show this graph sometimes. There was 1 month where I was aging at something like 1,300 times the rate of normal aging, because just based on the change in biological age. And yet, in that same month, my Dunedin pace of aging score from the same company, same test, was less than 1. It was like 0.67, right? So those numbers don't match up. I can't be aging 1,300 times as fast as normal and have a pace of aging, a slope, that's 0.67. Yeah. So again, I don't have any confidence that the consumer-facing Dunedin pace of aging number means anything more than the biological age that you get from the same test. I can tell you, you will bounce around in that Dunedin pace of aging. I've had probably 0.6 was my lowest, but I've had like 0.6 to 1.0. Never been above 1. So maybe that's a good thing. Maybe it's just random chance, but I don't put any faith in that number as a metric of anything relevant.
I see. Okay, then maybe that answers my question I was just about to ask, but do you think there's anything in particular that seems to correlate most with the score? Like, would a reduction in inflammation, HRV?
So again, you're making the assumption that the company selling the test is actually doing it right. Right? I mean, really, right? So yeah, I could come up with all sorts of things that I would think might impact that Dunedin pace of aging score, right? It also depends a little bit on how they're doing it. And this is where the technology is also changing and the understanding is changing. So originally, all of these tests were done just from blood cells, right? And immune cells in particular are going to dominate. Now people are starting to learn you get different results if you fractionate the cells within the blood that you're actually measuring the epigenetic changes in, right? So you may have seen, so some of these companies are now starting to roll out these organ-specific clocks, right? Yeah. So you can make a prediction based on where the cell type, the epigenetic signature is coming from on the rate of aging or the biological age of the organ or tissue of choice. Again, scientifically, super cool, super powerful. I actually am really excited by these organ-specific clocks. I actually think the proteomic and metabolomic clocks are probably better than the epigenetic clocks right now. So it's a different type of thing you measure. But the idea that from a single blood sample, we can measure function of multiple organs, really powerful, right? And in a more quantitative way than just typical blood chemistry. But all of that is to say, it does depend on how you do the measurement and whether you're looking at all of the blood cells together or you're fractionating out different types of cells, you will get a different answer. But again, this goes back to, we don't really know what these companies are doing or whether they're doing it well. And so, yeah, if you have less inflammation, or if your immune system's functioning better, or if you have less stress, or if you're eating a healthy diet, or if you're doing all of those things, I would expect your Dunedin pace of aging would be lower. if the company's doing it right.
I see. Okay. So one of the things, so I think the tests are really interesting. It's just what kind of claims are you going to make based on it? So I've seen some Instagram, Facebook ads from Brian Johnson where he's saying, uh, is your diet working? Should you do vegan? Should you do carnivore? Should you do Mediterranean? And he says, don't trust opinion, trust the data. You should buy this speed of aging test to see if your diet is working.
Okay. So I'm just going to say it. Nobody should make any change in their health or buy any product based on what Brian Johnson says. My opinion, and I feel pretty strongly about that. Okay, so let's not talk about Brian Johnson. Let's just I think unfortunately, people, there's a lot of misinformation out there. The tests would be great if they actually worked the way that the companies are claiming they worked, both for precision. And so precision is just, if you do the same test multiple times, how close together are the measurements, right? They should be identical, but everything's going to have some noise. Any measurement is going to have some noise. Accuracy is how close is it to the true value, okay? We don't know precision or accuracy for these tests at all. And I think what I have learned is it's really bad, just from the experiments I've done, at least the precision part. So people are making an assumption here that the tests are actually measuring something relevant. And the data we've got right now is that they aren't. And there's no way we can estimate accuracy of the test because we don't actually know what the true value is for biological age. So I would say a much better approach at this point would be to use biomarkers that we actually do know something about. We've got a lot of biomarkers that are really informative for health. Use those. Don't use a $500 test that isn't very good and you don't even know what it's measuring. Like, it makes no sense.
Right. It's like if you could just check your inflammation or your triglycerides, your insulin or your glucose.
Yeah.
We already have.
Or body composition, right? Body composition. Really useful tool. Really easy to measure.
Mm-hmm. Okay. Speaking of, because we touched on rapamycin a little bit. Let me see. Okay, there was this preprint that you had commented on in one of your videos that suggested that rapamycin may increase speed of aging based on its effect on these kind of biological age clocks.
Yeah.
Could you share with us a little bit how that worked and what's going on there?
Yeah. So first of all, this is a sort of observational study of people who they looked at a whole bunch of different lifestyle factors and drugs that people might be using. So it's complicated. It's a complicated data set in the sense that nobody's only doing one thing. All of these people are doing a bunch of stuff, and they're just asking, of all the people who are eating a Mediterranean diet or exercising regularly or taking rapamycin or taking this other drug. And then if we only look at the epigenetic clocks in those people, do we see any trends, right? So it's a limited, you have to be careful with those kinds of data, but that's still a useful study design. But you have to be careful for that reason. And then they just asked across these different factors, which were associated with a lower biological age and which were associated with a higher biological age using these clocks. I don't know for sure. My recollection from looking at that preprint was this was done through a company, True Diagnostic, did the tests. I'll just say, when I did my experiment, the True Diagnostic tests, the duplicates were 20 years apart. So people can take that for what it's worth. So there's some concerns there potentially about what the data actually means. But then I would also say, in that study, they didn't find a reduced rate of aging with a healthy diet or with exercise. So if the positive control doesn't work, that's kind of like the starting point for any experiment. If your positive control doesn't work, you can't conclude anything from that experiment. So I don't think we can conclude anything about any of the other interventions there. Take away all the nonsense about the lack of precision and accuracy in the tests. Even if we were as charitable as we could possibly be, and we said, we believe the tests work, I don't think you can conclude anything from the experiment because the positive controls didn't work. So that tells you either the design was wrong or you did something else wrong in the analysis or the experiment.
I see. So the result that they got from, in terms of the expected effects from a diet and lifestyle change, it was such an unexpected result that it suggests that the rest of it is just...
Either the tests aren't telling you what they're telling you, or the way the study was designed was not sufficiently powered or otherwise constructed to see a result that should be true. Again, if you want to say exercising and eating a healthy diet isn't good for your health, by all means, you're free to say that. I think we all know that's not true. And so again, I just don't think you can use a result for one intervention, rapamycin in this case, but you could pick any of them in that paper and have any confidence that that's a true result when the expected result for the stuff we know works for health didn't give a true result.
Okay, that makes a lot of sense. Maybe this would be a good time to talk about sirtuins and how you were the first person to discover that they had a particular effect on longevity. Could you walk us through that story?
Sure. Yeah. So this was actually my first project as a graduate student. So I actually went to graduate school thinking I was going to do structural biology, and I heard a talk by Lenny Guarenti, who was at MIT, about how his lab had just started studying aging using genetics and molecular biology. And I was immediately fascinated by that, joined Lenny's lab and started studying the genetics of aging. And in this case, we were working at that time in a single-celled model in budding yeast. And David Sinclair was in the lab at the time. Brian Kennedy had just left. And so we sort of had some ideas about how aging was working in yeast. And in conversations with David, actually, we got interested in this gene in yeast called SIR2. That's where the word sirtuin comes from. So when I started, that word hadn't been invented yet, right? So I was studying SIR2 in yeast. SIR2 stands for Silent Information Regulator, just as a sort of tie back. It's got that name because it repressed expression of certain genes through epigenetic mechanisms. So this was also the first epigenetic mechanism for regulating aging that was ever discovered. So in any case, I started studying SIR2. We knew that if you deleted SIR2, it would shorten lifespan. But again, there's lots of ways you can shorten lifespan that have nothing to do with aging. So I thought what we really need to do is turn it up, overexpress it, and see if we can extend lifespan. Because if you do that, you almost have to do that by affecting the aging process. So along with another graduate student in the lab, Mitch McVeigh, he and I created a set of strains overexpressing different levels of SIR2. We tested them. Turned out it increased lifespan by about 30%. So we were able to show a dose response where we could go up or down and modulate lifespan directionally to show that SIR2 overexpression slowed aging. And so that was cool. That was in yeast. We published that, I think, in 1999. People sort of paid attention, but people didn't get super excited about it until other people, one in Lenny's lab named Heidi Tissenbaum, and then Steve Helfand's lab in flies, showed that in C. elegans and in fruit flies, you could overexpress the corresponding gene and extend lifespan there. And then all of a sudden people thought, oh, this might be an evolutionarily conserved mechanism for slowing aging. And then about the same time, Shin Amai, and I was a co-author on this paper, but it was really Shin's work, discovered the enzymatic activity of SIR2 and then the human version, which is called SIRT1, sirtuin-1, to be an NAD-dependent deacetylase. So that's the epigenetic mechanism. The reason why I'm talking about this is that's how NAD comes into the story. So it's this NAD-dependent piece that sirtuins require NAD to do their epigenetic job. The cool thing there was this was the first time that particular chemical reaction had ever been described. So it was a brand new enzymatic reaction. So that the combination of sirtuins affecting lifespan across multiple species and this really new cool enzymatic activity sort of lit this fire. And that's where the name sirtuins come from. So sirtuins are the family of NAD-dependent enzymes that are structurally related to SIR2, sirtuin. Turns out humans have 7 of them, SIRT1 through 7. And so that's the sirtuin. That's how sirtuins got started in longevity. Then it has turned out to be this sort of wandering road and, all sorts of interesting stories. A lot of interesting stories there. I'll just say today, I think, despite hundreds of millions of dollars being spent trying to prove sirtuins are master regulators of longevity, it doesn't seem like they are. They are some of them. Again, I said there's 7 of them, not all of them, but some of them seem to be in this network that I talked about. They just don't seem to be particularly effective nodes in mammals, at least in mice and probably in humans as well, they don't seem to be particularly effective nodes that we can tweak, turn them up or down to affect lifespan. Having said that, it's really, really hard. I'm not saying sirtuins don't have any role in aging. And if you had to pick one, I'd say the data right now is SIRT6, which, we can talk about the mechanism if you really want to, but there's a little bit of evidence in mice that activating SIRT6 probably is able to somewhat extend lifespan. It's not as effective as rapamycin, but you can get some benefit on lifespan. So I think they're involved in aging, but they're just not particularly important, or maybe I should say as important to some other things that we're able to manipulate more easily.
I see. Are these downstream of mTOR in some way?
They're connected. So I would not say downstream at all. And again, it gets really complicated when you try to actually draw the network out because there's so many connections and so many missing pieces. So it is the case that you can find, 2 or 3 step removed connections, particularly between SIRT1, which is the one most people were interested in initially, and mTOR, and then another piece of the network called AMP kinase or AMPK. So those 3 seem to talk to each other in interesting ways.
I see. So you mentioned that SIRT6 seems to be the most interesting one. Is that the one that is kind of responsible for the hype around NAD supplements?
The hype around NAD supplements is interesting. So again, the hype is really built by a small number of people who sort of proselytize this thing, right? Again, it's the data and what's out in the public domain are very different around lots of the sirtuin space in general, and NAD precursors and resveratrol as well. So it was mostly through potential effects on SIRT1, but when SIRT1 turned out to probably not be that important, people kind of looked for alternative hypotheses. And so I would say I think separately about sirtuins and NAD in the context of aging. Are they related? Sure, because sirtuins use NAD for their chemical reaction. But I would say NAD affects thousands of other enzymes and chemical reactions in our cells, including sort of very fundamental stuff around energy production and carbon metabolism and mitochondrial function. So the idea that if NAD precursors have benefits in the context of aging, it's because of sirtuins, I think is not well supported. And it's more likely to be other roles of NAD. Just to give you an example, when you have some level of mitochondrial dysfunction, so NAD can exist in 2 forms, NAD and NADH. That's a little bit of a simplification, but one is oxidized, one is reduced. When you have mitochondrial dysfunction, it turns out that our cells tend to accumulate NADH, and that's problematic if you get too much NADH and not enough NAD because you kind of shuts down basic chemical reactions. So in the context of mitochondrial dysfunction, which we all experience at some level, putting more NAD into the system might be beneficial just because you're restoring that ratio of NAD to NADH, have nothing to do with sirtuins, right? There are lots of other explanations that could be important there. I think it's still an open question how important at a sort of population level is NAD decline or NAD dysregulation for aging in most people or most animals. I certainly believe there are a subset of people where NAD dyshomeostasis does become problematic and that the NAD precursors can probably have a benefit in that context.
Okay, so the people who need it more benefit more from it.
That's true of almost anything, but yes. Yeah. But I think the question is it, 0.5% of people or is it 95%?
Oh, yeah.
Well, we really don't know the answer at this point.
Okay.
I would say the other thing is with NAD precursors, the primary, the basic literature, so laboratory studies and the human clinical literature is very uneven. So there are certain areas of biology where the literature in that area is, for lack of a better way of saying it, a mess. So the NAD precursor literature is a mess. And by that, I mean, there have been multiple studies that have failed to be replicated, where an initial result was found and then people tried to replicate it. And so it's usually, I can, especially if it's in an area that I'm knowledgeable about, I can kind of look at the body of literature and develop a sort of, probabilistic opinion based on that body of literature and the number of times certain things have been reproduced or not reproduced. NAD precursors are really hard for me because there's just so much smoke. Like there's something real there, but there's also so much stuff that hasn't been reproduced. I don't know what the fire is at this point. And so I really struggle with the NAD precursor and making recommendations around NAD precursors for that reason.
I see. Okay. So you don't take it yourself?
I don't. And I mean, again, we can talk about the different NAD precursors, right? There's nicotinamide riboside, nicotinamide mononucleotide. Those are the two that are expensive that are sold. And then there's niacin and nicotinamide, which are cheap that are sold. They're all NAD precursors. They will all raise NAD if you take them orally. It's not clear that one is better than the other. And then the other thing that's become popular in, longevity clinics is NAD drips, right? So just putting NAD in through IV. Again, all of them are based around the idea of boosting NAD levels in circulation. So I don't take any NAD precursors regularly, in part because I'm not super convinced, but also there's been one study now in mice that NMN at the doses that have been reported to increase lifespan also cause kidney pathology in a subset of aged mice. And there's a, somewhat compelling potential mechanism for that has to do with metabolism of what we would call supraphysiological doses of NAD in the liver that lead to a byproduct called 1-methylnicotinamide that then has to be cleared by the kidney.
I see. Okay. What do you think about these gene therapies? Like, I know follistatin gene therapy is very hyped up at the moment. Do you feel excited about those, or do you think there's any utility?
So I think one general statement I would make is I think gene therapy as a therapeutic modality has a lot of promise, right? So the idea that we can, in a variety of ways, express different genes or change the genome has a lot of promise. It's also very technically challenging, and there are, as we've learned over the last 20 years, some risks that we have to figure out how to mitigate before we start doing this at scale in people. But certainly for rare diseases where there's a specific genetic cause and we can go in and fix it early, hugely powerful for those individuals. But there's been this sort of, I don't quite understand why, but this enthusiasm for rushing to gene therapy for longevity, right? So let's talk about the premise of that.
You mean like for average healthy people?
Yeah. For healthy people to enhance longevity, right? That's sort of the use case that you hear talked about. So let's think about the premise of that first. When would you want to use gene therapy for longevity in people? Well, I think at a minimum, you'd want to have a gene that you want to turn up or turn down where you're very confident, at least in mice, that it robustly increases lifespan, right? That's got to be the starting point. You got to have some reason to do it, right? So what are we messing with gene therapy, right? I'm not aware of any gene therapy out there right now where the data is particularly robust in mice that we would do this in mice and be 100% confident that it's going to have a positive outcome. Clotho is probably the closest one. And so if somebody was put a gun to my head and you have to do a longevity gene therapy today, what are you going to do? I'm like, give me Clotho. Okay. I believe it. Okay. Not suggesting anybody do that because I'm not, but that looks really interesting to me. Follistatin, it just makes no sense. So there's one mouse study that I think most people in the field don't really believe at this point that reported a lifespan extension. Hasn't been replicated. The mechanism doesn't make a ton of sense. So the idea that we should rush this to people, to me, seems like not a great idea. I know a lot of people are enthusiastic about it. I kind of get the idea. I mean, you look at like the bulls that have, I get the idea, the bro science, I get that. But from a longevity perspective, I don't think there's a lot of reason to believe that this is going to be beneficial. And given the way it's being done, and I totally like, I get why people are frustrated with the FDA. I am as well. Believe me, we could do a whole episode on that. So I get why people are like, we could never get this through FDA. Let's go do it offshore and let's get this to people, right? I understand that mentality. But when you do that, it's done outside of any sort of regulatory environment that ensures safety, right? Even in the manufacturing piece. And so again, it's kind of like these direct-to-consumer companies selling the biological age test. You have to believe these people who say, yeah, I've got this clinic on this secret island, and we do everything to the highest quality, right? You can trust us. You've got to trust them. And I don't know, maybe I'm cynical, but I don't. So I think there's that piece there. So there's both the therapy itself, and then there's, are these people doing it to the highest quality? How do you know? Who do you trust? Who do you believe? Neither of those line up for me as a great idea at this point.
I see. So two things on that. I think one of the appeals is the plausible deniability of people. They just want to get jacked, but then they can say they're doing it for longevity purposes.
Right.
And then the other thing is I had heard that if you really wanted to have a significant effect via this type of mechanism, it would have to be something that was present from birth, basically. So the modification to follistatin would have had to have been present from birth, or you're not really going to get a significant effect. Do you know anything about that? Or
so certainly, I mean, that is true for many genetic interventions that increase lifespan. So again, does follistatin increase lifespan? TBD. One paper, okay, we don't know the answer to that. But for many interventions that increase lifespan, yes, you get the biggest effect if you go through development with that change, like lower growth hormone, lower IGF-1, right? It also affects body size early on. In fact, it turns out that high growth signaling during development leading to larger stature. So when I say body size, I mean stature, not obesity, right? Larger stature is associated with accelerated aging across mammals, right? So you see this most in dogs. Think about a big dog versus a small dog, but it's true in other mammals as well.
So stature means like height and muscle mass?
Right. Body size, generally speaking, in a non-obese state. Yeah. So if you went through development with higher follistatin level, I'm not a follistatin expert, so I don't know what that would do to stature, but certainly it would make you more buff. My intuition is if that did anything to longevity, it would shorten lifespan. Okay. I think this idea of hyper, anabolism, or let's just say, higher muscle mass, I think being bigger because you have higher growth signaling during development in general, in people, it's not a huge effect, but in general, shortens lifespan and accelerates aging. Maintaining muscle mass later in life is a pro-longevity benefit, right? For metabolic reasons and for bone density and resistance to frailty, right? So with any therapy, and again, resistance training is a therapy that works. Like that works, right? Doing that later in life so you maintain muscle mass and functionality, we know is beneficial. Follistatin, I don't think we have enough data we definitely don't have enough data in people to know, how many times do you have to do the therapy? How fast does it go away? Would repeatedly, let's just say it worked the way you wanted it to, would repeatedly administering systemic follistatin, preserve muscle mass better during aging? It might. Would it have other effects on like your heart, mostly muscle? Is that a great thing? Maybe not, right? So could there be adverse consequences we haven't anticipated? Yeah. And then these repeated gene therapies, people, I'm all for vaccines. Like I'm not a vaccine denier at all. Vaccines work, but I also think there are unanticipated consequences to repeatedly injecting genetic material into your body. Every time you do that, it affects your immune system. It causes an inflammatory response. If that genetic material is not cleared, it can create a persistent inflammatory response. A persistent inflammatory response can suppress your immune system's normal functioning and increase your risk of developing cancer. Could that happen with repeated or a single dose gene therapy? It might. Like I would be a little bit concerned about that. So I think we just don't really know what the long-term consequences are. And that's why I say, again, I think gene therapy as a modality has a lot of promise, but I would really want to be sure that whatever's in that gene therapy is very likely to have a beneficial outcome. And I don't think we know that for follistatin, and have some better understanding of what the long-term consequences are going to be.
What made you say Clotho would be the one you would do if you were forced to pick?
I mean, it's a super interesting hormone, peptide hormone that is in mice been shown to increase lifespan. It goes down with age. And I think particularly the work from Dina Dubal is really, really compelling in this space about the ability when you raise levels to have a positive impact on cognitive function, and the potential that, in dementia, it's because at least associated with and potentially causally associated with declines. So I think the cognitive data is best, but it seems as if it has a systemic aging effect as well. And again, we don't know in people yet. In fact, I think Dina's involved in a company that's trying to develop this as a therapy. I don't know if it's a gene therapy or an injectable. I haven't really followed that part, but I'm pretty intrigued with that molecule as a potential therapeutic.
Interesting. Are there any other niche therapies, niche interventions that you're excited about or interested in?
Yeah. Well, I mean, I think the epigenetic reprogramming stuff, which we touched on earlier, is probably the one right now in the field that has the highest potential upside, given what we know, right? So again, this idea that if we could optimize reversal of the epigenetic changes that are associated with age, that we could get large effect sizes on lifespan, or if not lifespan, at least potentially multiple health span metrics. So to me, that seems like the one that is farthest along, that has the potential highest ceiling. Again, I think much like gene therapy, the actual implementation of that in humans could be very challenging, because it's the kind of thing where if you push it too far, you potentially do really, really bad stuff. So the risk of very bad outcomes is higher than it is with something like rapamycin, right? Where at least acutely, you could take 100 milligrams of rapamycin and nothing's going to happen to you. Don't do it. I'm not suggesting you do that, but at worst, you're going to get high lipids, right? Whereas if you overdo epigenetic reprogramming, you're going to get crazy cancers, or you might just turn into a bag of stem cells, which is probably not a good outcome. So, but I'm interested to see how that evolves. I think the other area where there's lots of enthusiasm, and I mean, reason for enthusiasm, but I don't think nothing I've seen in this space is, at least in laboratory animals, better than rapamycin, is around what people call senolytics. So this idea that you can specifically target and potentially clear senescent cells that accumulate in the body with age makes sense. And I think that it should be doable, and will probably have benefits, certainly for, a variety of different age-related diseases. I just don't think the effect size is going to be all that much larger than something like rapamycin.
Okay.
But it might have a better side effect profile.
I might have asked you this already, but so rapamycin is basically like the best molecule, or the most evidence molecule for increasing lifespan, compared to whatever metformin, NAD, resveratrol.
So in laboratory animals, yes, for sure. No question about it. Hands down, most robust and reproducible molecule for slowing, I would say extending lifespan, but also across many, many different tissues and organs, slowing age-related decline, and at least in a few cases, reversing age-related decline. So in the heart, the brain, the ovaries, the immune system, and the oral cavity, people have shown that you can take an old mouse where there's already functional decline, start treatment with rapamycin, and within 6 to 12 weeks, actually see an improvement in function. Which again, from a translational perspective, is really powerful if it works the same way in people. Again, we just don't have the clinical trial data in people to say with confidence that rapamycin works the same way in people. And I know a lot of people are concerned about the side effect profile. I think that's largely because of the way rapamycin was developed as an organ transplant drug. So high doses, daily dosing, sick people taking immunosuppressants, right? Very different situation than off-label use of rapamycin. But the truth is, because we don't have double-blind, randomized, placebo-controlled clinical trials, and we don't have decades of data on people using rapamycin off-label, we don't know what the long-term side effect profile is. I think it's become clear to me, at least, that the short-term side effect profile is pretty minimal at the doses most people are using rapamycin off-label. A couple other comments to make. So you mentioned some specific other molecules there. So we talked about the NAD precursors already. Those have not been reproducible in mice for lifespan. So I think the jury is still out whether they actually increase lifespan in mice. Resveratrol is the most debunked anti-aging molecule in history. There had been a meta-analysis with, dozens, if not more than 100 studies that looked at resveratrol for its effect on lifespan across many, many, many different organisms. The net effect is 0. What that means is among published studies, remember there's a publication bias towards positive effects. Among published studies, every time there was a positive result that increased lifespan, there was a negative result that shortened lifespan. So the average is at 0. And resveratrol was never shown to increase lifespan in mice. That was a myth that was sort of propagated through, I don't know, let's not say, I don't have to say, well, I won't say it, through misrepresentation of the data. Whether that was intentional or unintentional, we can leave that out, but it was misrepresentation of what was actually shown.
Was that misreporting in terms of someone was saying something, but there was no study to back that up, or they were misinterpreting a study, or the study was fabricated?
They were, yes. So I should clarify. So the original study, which got a lot of attention because this is what led to the whole idea that red wine is good for you and is a longevity medicine, right? Because red wine has a little bit of resveratrol in it. So the original study that led to that, which was published in Nature, I think in 2013, 2011, can't remember, looked at mice on a very weird high-fat diet. So it was a high-fat coconut oil diet, which is never really used in the literature. I don't know why they picked that diet, but it's a very weird diet. The mice got extremely obese. They got so obese that they developed fat, a lot of fat around their internal organs. It's at least been said, I don't know if it's actually true or not. It's at least been said what they actually died from was the inability to breathe, to bring in enough oxygen. So there was so much fat around their internal organs. But anyways, they were massively obese on this weird diet. In that context, resveratrol did reduce the obesity and they lived longer. But they lived longer because they weren't dying from this massive obesity. On the mice that had a normal diet, there was no effect. That was, presented to the media as if resveratrol slows aging and people should drink lots of red wine. Then kind of the gold standard for mouse longevity studies, which is the interventions testing program, tested resveratrol and also found no effect on lifespan in mice. So I think it's pretty clear resveratrol does not increase lifespan in mice. And when you look across all of the experiments that have been done in every organism where it's been tested, the net effect is 0. That doesn't mean resveratrol can't increase lifespan sometimes in worms. Maybe it can if you do it just right, but it is certainly not a consistent longevity drug. And there is no reason that I'm aware of to believe that resveratrol modifies the biology of aging in any positive way.
Wow. So wasn't that the proposed mechanism behind the so-called French paradox of they eat so much saturated fat, but they don't have heart disease? It must be something about their diet. Maybe it's the red wine. Yeah. And then if I'm remembering correctly, I think it was specifically them saying, oh, we think this molecule. Yeah. Is that right?
I mean, you're right. Some people did say that. Okay. Again, unfortunately, you see this all the time. And I mean, it's a hypothesis, right? It's an interesting hypothesis. Probably testable. But it's also wildly speculative, right? There's lots of other things. First of all, is the French paradox even real? Like, I think there's questions around that, around the blue zones in general. Okinawa, absolutely, I believe that. The other blue zones, I don't know. There's maybe some reason to doubt. But is the French paradox even real is question number one. And if it is, there's probably lots of things that could lead to that. But I think alcohol is a different sort of question. So it is interesting when you look. And there's a little bit of controversy about this, but I don't think the data is all that controversial. When you look at a bunch of different population studies and you look at alcohol consumption as related to all-cause mortality. So all-cause mortality is a function of alcohol consumption. So it's correlative, okay? Correlative. It's a J-shaped curve. And what I mean by that is, it looks like a J when you plot it. You start out at a certain level of mortality if you don't drink anything at all, right? Your risk of dying is a certain number, right? If you drink a little bit, it actually goes down. There's a little dip. And then once you start to drink more and more and more, it goes up. That's the J shape, right? So that seems pretty consistent for alcohol writ large. Now there's a little bit of an effort. It seems to me like it's more propaganda than science, but there's a little bit of an effort to demonize alcohol and say all alcohol is bad. But at least for mortality, there really is this little bit of a J-shaped curve. But that doesn't seem to depend on the type of alcohol you drink. So there's no reason to believe that's red wine specific or resveratrol specific. Again, you can come up with all sorts of explanations. Maybe the alcohol itself has an effect on particularly cardiovascular disease. Brian Kennedy's done some work on alcohol in mice and can actually find a dose of alcohol that increases lifespan a little bit in mice. Maybe it has to do with the social environment, right? The fact that if you're not drinking alcohol by yourself in front of the TV, you're usually doing it with friends and family, and this human connection piece is important. So that might be part of the correlation. The answer is we really don't know, but I think it's pretty safe to say it has nothing to do with resveratrol.
I see. What do you think is the most enticing piece of evidence for the longevity benefit of socializing? I remember Robert Waldinger had a very interesting talk on this Harvard study that followed people for decades.
Yeah. Yeah. So I mean, again, I first of all, this is outside my area of direct expertise. So I'm not an expert. There are people who know way more about this than I do. There have been multiple studies that have shown that in a variety of different contexts, human relationships, I'll just put it broadly under that, are associated with better health outcomes and lower all-cause mortality. And, I think those human relationships can take a variety of forms. I think the ones where I've seen the biggest effect and probably the most consistent data are spousal relationships. And the reverse also is true. Bereavement is associated with, or death of a spouse is associated with an increase in all-cause mortality, right? Often. So, but again, is that because of the social interaction, or there are other reasons why having a spouse could also be a good thing? You fall down and get hurt, somebody's more likely to find you than you lay there for a week, right? So proving causality when it comes to this social piece is much harder. And of the 4 pillars I talked about, eat, move, sleep, connect, the connect pillar is the one where there's the least sort of hard biochemical molecular data. It really hasn't been studied. It's an area where I would like to see more effort put. I would also like to see more in sleep. I think sleep is an area where now people are starting to look at the mechanistic connections, but we really don't have a great understanding at a mechanistic level of how sleep connects to the biology of aging either. Back to the social piece though, one of the things that I find interesting, given my interest in dogs and the Dog Aging Project, is there do seem to be even physiological benefits from a human interacting with a companion animal, on both the companion animal and the person, right? So people have looked at several physiological biomarkers, including stress levels, and seen that humans interacting with, I think it was mostly in dogs, but I assume this would be the same in other companion animals, actually lead to reductions in stress and improvements in physiological biomarkers. So I again, it's a little bit speculative, but I'm going to go out on a limb and say that, you can get benefits from interacting with your companion animal as well.
Do you think, is that mediated mostly by oxytocin, or?
Certainly that's part of it, right? Again, this is where I say, I feel like the actual hard science and biology and neurobiology here still pretty limited. So I believe that's part of it, but I would also say often in science we go after the easy hypotheses. So that's an easy hypothesis. It makes sense. It's probably part of the story, but is that really the whole story? Is it the most important piece? I don't know.
Are there any particular protocols you've seen that, I know there's a couple different people doing different, you got like Julie Gibson Clark, of course, Dave Pascoe, there's Sim Land. Was there any particular, let's say, protocols you saw that you were excited by?
Yeah. So first of all, I'm not a big consumer of biohacker protocols in that sense, like trying to, figure out what's the best stack. And I can tell you why. I think Seam, of the people who I've interacted with, I think he certainly has the same foundation that I do, right? When you see him put together his list of 8 or 10 things, lines up perfectly with what I think about is where people should start, right? So, but I tend not to apply complicated protocols in my own life or try to identify complicated protocols that I think are going to be optimal. And part of the reason for that is I take a sort of just maybe a different philosophical approach. And I would put myself in the biohacker category. I've done my fair share of biohacking, but I try to do one thing at a time to try to see what changes and what doesn't change. Part of the reason why I don't throw everything at the wall, take a stack of 50 different supplements, is because, first of all, I don't think we have any understanding of combinatorial interactions. Again, that's part of the rationale for the WormBot. We can't predict when you do 10 different things to your body how that's going to interact and impact. And So first of all, I don't think there are 25 different supplements I feel really great about anyways. But if I did, I don't know that I would put all of them together and just assume that the sum is going to be greater than the parts or even as good as the parts. Part of the reason I say that is I think of our bodies as, an intricately complicated piece of machinery. Think about the most complicated piece of machinery you can imagine, right? And a supplement is kind of like going in there and randomly unscrewing something or pulling a wire out and putting it somewhere else, right? Maybe not random. Hopefully there's a little bit of logic to it, but kind of random. So if you do that with any complicated piece of machinery and you move 25 things around, what's the likelihood you're going to make that thing function better? Pretty low.
Right.
What's the likelihood you're going to make it function worse? Pretty high.
Yeah.
So, I mean, that's maybe a little bit of an over exaggeration of the risk reward, but I do feel like in the absence of any data about how these things are going to interact, you're more likely to impair function than make it better. So I prefer to be as much as possible biomarker guided and as much as possible functional biomarkers, strength, VO2 max. I would put body composition sort of under a functional biomarker. But also blood-based biomarkers that we're pretty confident are predictors of health, and then use that to kind of guide how I optimize my lifestyle, any supplements, any pharmacology that I'm doing.
Right. Is there anything outside of those pillars we were talking about that you're interested in, like sauna, infrared light?
Yeah. I mean, I think sauna is super interesting. The data I've seen, again, is it going to be broadly beneficial for most people? I don't know, but the data I've seen, it looks pretty good. The biological mechanisms are plausible. I don't right now regularly do sauna or cold plunge, but I could see myself doing it. If I had a sauna in my backyard, I'd probably do it. Right. So, but I would also say there's nothing other than hormones. And I think a lot of people should look at hormones, especially once you get above 45, that I would put outside of the direct lifestyle pillars that I would put in the kind of big mover bucket, right? There are levers you can pull that will have a big impact if you fix them. And I think for a lot of people, hormones would be in that bucket. I can't really think of it. Maybe I'm just not thinking of anything, but I can't really think of anything from an interventional perspective that I would put in that likely to have the same level of impact. But then I think, next level down, what might you think about? I mean, maybe vitamin D and omega-3, just because you can measure and we kind of know what the ranges are. And I like things that you can measure, supplement, measure again, and make sure you're there. But unfortunately, with a lot of these supplements, we don't have that. We don't know what the optimal range is. So we can't, even if we could measure it, we don't know. So that's where I try to be guided by biomarkers. So kind of next level down, I put rapamycin in that bucket for myself personally, right? And I can just say for myself personally, the first time I took it, I was dealing with what's called frozen shoulder or adhesive capsulitis, really painful, couldn't throw a ball with my son, couldn't sleep. The doc said, go back to physical therapy. It might go away in a year. And I was like, fuck this. I can't do this for a year. That's why I took rapamycin the first time. And within 10 weeks, it was gone. Could it have been placebo effect? Sure. Whatever the case, it impacted my quality of life quite substantially. So, and then I've taken it multiple times. I cycle it. I've taken it multiple times since then. I've never had anything that profound, but every time I do, I have this perception that the aches in my wrist and elbows from resistance training go away. Okay. Is it real? Is it placebo effect? Again, I don't know, but the biological mechanism is plausible enough from an anti-inflammatory perspective. I think it's probably real. I'm pretty sure it was real with my frozen shoulder. So regardless, so I put rapamycin for me in the bucket of that was a big lever because I had this acute thing that was fixed. And I think for some people who have high levels of chronic inflammation, sterile inflammation in particular, a cycle of rapamycin, and I've heard lots of anecdotal stories, can have an impact. But that's sort of an individual thing. I'm not suggesting anybody should take rapamycin, and certainly not suggesting everybody should.
Right. So you're not on it consistently. Like you right. I guess you cycle it.
I usually do 3-month cycles. Yeah. I mean, I'm not, it's funny. I'm very much a scientist in my head, but in my application in real life, I don't always take the most scientific approach. I've tried to get better at that, but yes, most of the time I've cycled it for 3 months on. Oh, okay. Then I usually take 3 to 6 months off.
You didn't notice any side
I have not noticed any side effects from rapamycin personally. I know about 15% of people get canker sores. So we actually did a study where we collected data from 333 people who were using rapamycin off labels. So not for organ transplant, but for potential health benefits. And we compared it to 100 and some people who'd never used rapamycin. And the only side effect that was statistically significantly greater in the rapamycin users was the canker sores. 15% of people compared to 3 or 4% in the non-users. There were 6 other things that were different statistically, but they were all lower in the rapamycin users. And it was interesting. It was things like eye pain, abdominal pain, depression, anxiety. So who knows? There are, again, this is the hard part with mTOR and rapamycin. You can look in the literature and find a connection to anything you want to find a connection to. If I wanted to cite a paper that says rapamycin is good for depression, I can go find that paper. But do I really believe it? Yeah, not yet, right? So, but it was an intriguing sort of, blip that might be real.
Was the incidence of canker sores dependent on the dosing, or was that looked at?
Our data wasn't big. We don't have a large enough data set to really be able to address that, or I would say a quantitative enough. I mean, this was all survey based, so take it for what it's worth, right? It's a limited type of data to draw strong conclusions from, and that's why I'm always careful, when I talk about it. And not everybody. A lot of people are kind of like me, right? They might have done 6 mgs a week this month and 10 mgs a week next month, and then they take a few months off. And so it was very noisy from that perspective. So really what we could do cleanest was say users versus non-users. Yeah. So anyways, going back to supplements. So I kind of put rapamycin in that next level down. The other supplements that I mentioned vitamin D and omega-3. I usually haven't for a while because I'm, again, not super scientific or disciplined about it, but I don't think a multivitamin's a bad idea, right? You think of it as an insurance policy. All of these things when you're dealing with supplements though. So prescription medications are a little bit different because they have to be manufactured in, clinically approved manufacturing facilities, right? Again, compounding pharmacies are a different beast, but let's leave that out of the discussion for right now. If you're buying it from a reputable pharmaceutical company, they have to be manufactured to a certain quality. That's not true with supplements. So any supplement kind of baseline is do your best, consumer beware, but do your best to buy it from a reputable company that at least does third-party testing so you can have some confidence that what you think is in there, and there's not a bunch of other crap in there that might be harmful for you. That's actually another reason why people might want to be careful not to take 20, 30 supplements though, is a lot of these supplements have impurities in there that can accumulate when you start to take, a large number of them every day.
Like lead, right? Cadmium, heavy metals.
Exactly.
Okay.
Yeah. And I mean, sure, you can measure some of that stuff on a blood test, but still, just want to know, you want to be careful that you're not overdosing yourself on metals. Oh, well, so let's finish the supplement. I thought of one other experimental sort of therapy that I'm kind of bullish on and excited about recently as well. So for me personally, I think creatine makes sense. Again, it gets, kind of goes up and down in the hype cycle, but I feel like it makes sense, especially if you're doing resistance training, probably not a lot of downside and maybe some other benefits associated with it. So that's pretty much what I do. I think, other things that are interesting, alpha ketoglutarate, urolithin A, spermidine, NAD precursors. I kind of put those in the category of, intriguing from a longevity perspective. Rock solid glycine would be in there as well. Some evidence in mice, not always reproducible, some interesting anecdotal data in people. So that's kind of where I land on those things. And then there's a bunch of stuff I just, I don't know about. I haven't tried. Like, I don't pretend to be an expert in every possible, intervention that somebody's trying to sell you, right? I try to be guided by where's the most scientific evidence. Okay, so you want me to, I got 2 things now, actually, I'm excited about. Yeah, so 2 longevity things that I'm intrigued by and I'm going to try personally at some point. So one is therapeutic plasma exchange. Okay. So this is based on the idea initially of parabiosis, or what's called heterochronic parabiosis. This is the idea you surgically connect a young mouse to an old mouse so they share the same circulatory system. When that's done, what you see is that the old mouse gets healthier and the young mouse gets less healthy. And it's at least consistent with slowing aging or accelerating aging. And this has been seen over and over and over. You can even get a slight lifespan extension in the old mouse when you do this. That data is a little bit complicated because just the surgery itself, as you might imagine, shortens lifespan. So is it a real lifespan extension? In normal mice, nobody knows, but it's intriguing. And the hypothesis is there are both factors that accumulate in the aged circulatory system that are detrimental. And so you're diluting those out in the old animal. And there are beneficial factors in the young mouse that are helping the old mouse, right? So that seems pretty plausible. And people have tried to identify those things and there are candidates, but nobody really knows what the specific factors are. So therapeutic plasma exchange accomplishes half of that equation, right? So basically you're diluting out the bad stuff in the old blood, at least theoretically, right? And so the idea is you can go into a clinic, it's a couple of hours, you're hooked up by IV, they replace some of the stuff in your plasma with albumin and saline, right? So you're basically just diluting out some of the bad stuff.
Is that the total plasma exchange? Like
same idea. Yeah.
Okay.
Yeah. I don't know what's being done in the current version of the therapeutic. There's a few flavors of this, and you can do different things as part of the therapeutic plasma exchange. You can run the plasma through filtration columns to potentially pull stuff out. In theory, you could even put stuff in there if you wanted to, but the kind of basic version is just thinking about it as diluting out the plasma, right? So why might that be beneficial? Well, it could be beneficial for the heterochronic parabiosis effect, right? But think of the other stuff that might be in circulation, microplastics, metals, right? So I think it's intriguing. And for the microplastic perspective, I think it's really intriguing because I haven't yet heard of anything that might help get rid of the microplastics in an effective way. Certainly stop drinking out of plastic bottles as much, that stuff, try to avoid it. But how do we get rid of the stuff that's already there? This is intriguing to me, especially the idea that you do it once. And again, this is very speculative. So folks, I don't know. This is just speculation. But it's intriguing to me, the idea you might be able to dilute the stuff out, metals, microplastics, other toxins that are in your blood through one round. And then you wait a few weeks and maybe you can actually leach some of the stuff that's in tissues back into circulation just from exchange, right? And then do it again. So I'm intrigued and bullish on it. And I think there's also accumulating data in the sort of mild cognitive impairment area where therapeutic plasma exchange could have beneficial effects on progression to dementia. That's also really interesting and intriguing. So that's one therapy I'm enthusiastic about. And again, the mechanism lines up with my understanding of the biology. The other is this protocol that a guy named Greg Fahey developed called TRIM. So this is a thymus regeneration protocol. And so the idea is our thymus undergoes atrophy pretty early in life, maybe around adolescence or in your early 20s. And it shrinks in volume by about 50%. Your thymus is important for immune function and particularly generation of T cells. So you have a decline in the production of new T cells with age, and it continues to go down with age. And what Greg's idea was if you could regenerate the thymus, that could be beneficial for aging by regenerating immune function, right? And maybe helping to quell some of the chronic sterile inflammation that goes along with aging. So he developed a protocol. It's really simple. It's a combination of growth hormone, DHEA, and metformin. Okay. It's really the growth hormone that's doing the work here. And you don't have to do it for the rest of your life. You can do 3 months, 6 months, right? So the growth hormone is really what's doing the work. And it's doing two things at least to the thymus. So one of the things that happens during thymic atrophy is there's infiltration of fat into the thymus. So about 40 to 60% of the thymus by volume appears to become fat or adipose. The growth hormone is reversing that. Okay. So you remove that fat from the thymus, and then it seems like the thymus sort of restores volume, and maybe that's restoration through stem cells. I don't think we really know. The DHEA, I'm not so sure exactly why that's there, except it's a hormone precursor and it goes down with age. And the metformin is supposed to offset the negative effects of growth hormone on glucose metabolism. So it's supposed to just be a protection against that.
So the growth hormone isn't like injected into the thymus.
No,
it's in the
it's
circulation.
systemic. Yeah. Right. So I'm intrigued by this, and so I want to give it a try. I might substitute an SGLT2 inhibitor for metformin. That's a different type of anti-diabetes drug, in part because metformin can blunt benefits of exercise. At least there's some data to support that. And SGLT2 inhibitors do seem to increase lifespan in mice. So they seem to have a longevity effect, at least in mice. So anyways, but when I do it, I really want to do it properly. I want to get an MRI measurement of my thymus before and after so I can see if the volume changes. I want to figure out what are the right markers from an immune functional perspective to look at to see if thymic function is restored, and what other biomarkers does it make sense to look at. But again, the data that I've seen, it's got lots of issues. It's not a perfect study. That's what I mean by it. It's not a perfect study. It's like our rapamycin study. The best they could do in the situation, but limited in how strong you can feel about the results. But intriguing. So I want to give it a try and see. And again, I always think about these things through a risk-reward perspective. So what's the real risk here? People get scared by growth hormone. I think most of that is just, because it's been presented in a way that overstates the risks, particularly if it's only like a 3-month or a 6-month regimen of growth hormone. There are lots and lots and lots of docs who have been prescribing growth hormone to elderly people for a long time. Does it increase the risk of cancer a little bit? Maybe. Are people dropping dead from growth hormone? No. So I don't think growth hormone is a longevity drug, at least not, if you take it for years and years, but I also don't think it's very risky. DHEA is a supplement you can buy on Amazon, probably not very risky. And metformin or SGLT2 inhibitors, many people be taking these for decades. So probably not very risky. So I think the payoff from learning whether this actually rejuvenates my thymus is worth, 3 months or 6 months of giving it a try.
Yeah, cool. That sounds really interesting. I'd love to. Are you going to make like a video?
I will absolutely document that process.
Oh, great.
Yeah.
Okay. You mentioned metformin having a negative effect on growth of the muscles and that was one thing I noticed with Peter Attia, is he was mentioning that he was worried about fasting, reducing muscle mass too much.
Sure.
And so he had a pretty aggressive fasting protocol, if I remember correctly, doing like a 5-day fast every single quarter.
Yeah.
And me personally, I thought maybe that wasn't the fairest criticism considering he was on metformin at the time.
Mm-hmm.
So
yeah.
What do you think about
So
that?
I would separate those two things completely, first of all. Mm-hmm. So metformin is pretty easy to talk about the data because there's not very much of it when it comes to exercise, right? So what do we know about metformin? So we know metformin is a great anti-diabetes drug. It's a mitochondrial inhibitor. That's probably how it's working because when you inhibit the mitochondria, you activate AMP kinase, and that's the anti-diabetes effect. Outside of that, we know in at least a subset of men, and I don't know what the percentages are, but it seems like it's a relatively high percentage. A subset of men will see reduced testosterone when they take metformin. That could account for impacts on muscle mass. But then also there's been work, I think Ben Miller at Oklahoma was the original, or OMRF was the original one to show this, but now other people have seen this and even some human data. Metformin seems to, at least in some people, blunt some of the beneficial metabolic and hormonal responses specifically to exercise. So some of the signals that get produced by exercise that we believe are beneficial seem to be blunted by metformin. I don't think the mechanism is understood there, but as somebody who exercises regularly, I would be concerned about taking metformin. I mean, if you have diabetes, it's a great diabetes drug and you probably want to take it in that context. But if you're metabolically healthy, I wouldn't take metformin for a potential longevity benefit. I guess we haven't said this yet. Metformin does not increase lifespan in mice. Again, that has been widely misinterpreted. Does it improve some health span metrics? Probably. Does it robustly increase lifespan? Definitely not. Is it possible to get a tiny effect on lifespan in mice? We're talking like 3 or 4%, maybe, but it's not anything I would put in the category of a longevity drug. So people are just misunderstood, misinformed about that. So I don't think metformin is a great candidate for a longevity.
Oh, really? Okay. Yeah. Because there seemed to be so much hype about it.
Well, I think part of the reason is a small number of people who have a large podium who have said metformin's a longevity drug. Part of it is there was a study that was published several years ago now looking at a human population, and they compared diabetics taking metformin to diabetics not taking metformin. Diabetics taking metformin live longer than diabetics who don't get metformin. So in diabetic populations, metformin reduces mortality. And then they did a comparison and tried to compare it to a matched population of non-diabetics, so healthy people who don't take metformin. So the question is, if you compare diabetics on metformin to non-diabetics who are not on metformin, what's the impact on mortality? And that study reported that metformin in diabetics, those people actually had lower mortality than non-diabetics, normal people not taking metformin. That got a lot of attention, and I think rightly so. People were intrigued and enthusiastic about the possibility that metformin might increase lifespan or reduce mortality in people based on that. The problem is that study had all sorts of methodological flaws, and in subsequent studies in other populations, that result has not been reproduced. So in subsequent studies, diabetics taking metformin don't live as long as non-diabetics not taking.
Oh, so the study was decently replicated.
No, it was not.
Oh.
It failed to be replicated.
Oh, I see.
So that's partly why I think in the longevity science community, people are not particularly optimistic about metformin as a longevity drug. To be fair, there is correlative human data that diabetics taking metformin are at lower risk for a bunch of other age-related diseases, right? Cancers, cardiovascular disease, dementia. It's not perfect, and there are some contradictions, but in general, diabetics taking metformin are healthier than diabetics who don't take metformin and have lower risk for a bunch of age-related diseases. So if you want to make the case that metformin can impact heart disease and dementia and cancer, you can make that case. I don't know of any data yet that metformin is beneficial in people who are not metabolically perturbed at protecting them against those diseases. It might be. I have mixed feelings. You might have heard of the TAME trial, targeting aging with metformin. That's this big trial. I have mixed feelings about that trial. I would like to see it done just to get the answer, but it's also a really expensive trial, and I would put the money elsewhere if I was the money czar in the United States for research.
Yeah. Okay. About how much do you remember the estimated cost of it?
I want to say it was like 500 million, but I could be a little bit off. Maybe it was much less than that. I could be a little off. Certainly in the tens, if not hundreds of millions of dollars.
Okay. Interesting. Okay. Was there anything, excuse me, I think I covered all the things I prepared. Was there anything that you wanted to mention that I missed?
Not really. I mean, I think we've covered a lot. I think this has probably become clear from the conversation, but I think it's worth, reinforcing to people this idea that I would say a couple of things. One is the biology of aging is really biology. And what that means is it's a biological process that we can study and understand mechanistically. And as we do that, we have better and better opportunities to modulate that biology in a way that will keep people healthier longer and probably live longer as well. And it's, in my view, the most effective way to do that. It's much more effective to slow aging than it is to wait until people are sick and try to cure their diseases. So I just want as many people as possible to understand that concept and also recognize that we have been doing exactly the opposite for the last 50, 100 years in both biomedical research and medical practice. Our entire biomedical enterprise, from basic research to drug development to clinical practice, has evolved culturally around the idea that we treat people when they're sick. We wait until they develop a disease and then we try to cure it. It's much harder to cure somebody's disease after it's become a pathology than it is to keep them healthy in the first place. And yet still, and I think everybody would agree with that, yet still, 99.5% of the NIH budget goes to reactive disease research. 0.5% goes to targeting the single greatest risk factor for all of these diseases, which is the biology of aging. That needs to change. And in the healthcare world, I don't know what the numbers are, but it's probably not much different if you look at the expenditures towards treating sick people versus the expenditures towards keeping people healthy. I think that needs to change as well. So I hope that message resonates with people. And I think the more people who start saying those words, the better.
Yeah. Yeah. I think it really puts into perspective, because I think some people have the impression that, well, if we could significantly slow aging, that would have been done by now. But based on the budget allocation you're talking about, it kind of sounds like we haven't even tried.
Haven't even tried. In fact, I would say the advances made in the field of aging biology, or what people now call geroscience, which is just the science connecting aging to disease. The advances made in our understanding of that biology over the last 30 years are actually really impressive, considering the very, very limited amount of research dollars that has gone to that process. And if we were to actually allocate resources appropriately, I think we could make huge advances in a very short period of time.
Right. Speaking of which, so I remember seeing your title. It was like biologist and biogerontologist. Was that accurate for you? It's like Dr. Matt Kaeberlein, biogerontologist.
Maybe. I mean, so again, I think one of the challenges we have in this field is figuring out what are the right words to use, right? I mean, really, it's true. Gerontology, geroscience, biogerontologist, right? Those are not good words for the average person, right? Longevity is great. The problem is longevity has been co-opted by a bunch of people who don't know anything about longevity. And so now it means something different, at least than what I think of when I talk about longevity. I still like health span as a word. Yeah. Again, appreciate health span as a concept, though. It's not something that we can measure quantitatively, but I think we all understand what it means, right? Let's just maximize health span. Great. I don't think anybody can disagree with that.
Great. I think that's...
So I'm I don't know, health span advocate. How's that?
Okay, that works. Yeah. Great. Okay. Well, Dr. Kaeberlein, thanks so much.
Thank you. It's been fun.